Okay, so this is Chapter 8, an introduction to metabolism. An organism's metabolism transforms matter and energy subject to the laws of thermodynamics, and that's what we're going to spend a lot of this chapter focusing on. The living cell is a miniature chemical factory where thousands of reactions occur. Cellular respiration extracts energy stored in sugars and other fuels, and cells then apply this energy to perform work. Some organisms can even convert energy into light, as in bioluminescence. The ocean can glow and glitter like stars in the sky thanks to a natural chemical process known as bioluminescence, which allows living things to produce light in their body. Marine creatures like some fish, squid, tiny crustaceans, and algae produce bioluminescence to either confuse predators, attract prey, or even lure potential mates. We can witness this natural phenomenon when there's lots of bioluminescence in the water, usually from algae blooms of plankton. The bioluminescent sea will glow when it's disturbed by a wave breaking or a splash in the water at night. The totality of an organism's chemical reactions is called metabolism. Metabolism is an emergent property of life that arises from interactions between molecules within the orderly environment of the cell. Metabolic pathways begin with a specific molecule, which is then altered in a series of defined steps to form a specific product. A specific enzyme catalyzes each step of the pathway. And remember, a catalyst is something that speeds up a reaction. So here you can see an example. You have a starting molecule, which is acted upon by an enzyme, and reaction 1 takes place, producing B. Then a second enzyme is involved, producing reaction 2 and product C. And then that product C undergoes a reaction that is facilitated by enzyme 3, producing D, the product. Catabolic pathways release energy by breaking down complex molecules to simpler compounds. A major pathway of catabolism is cellular respiration, in which the sugar glucose is broken down in the presence of oxygen to form carbon dioxide and water. Anabolic pathways consume energy to build complicated molecules from simpler compounds. They are also called biosynthetic pathways. The synthesis of a protein made from many amino acids is an example of anabolism. The energy released by catabolic pathways can be stored and then used to drive the anabolic pathways. Energy is fundamental to all metabolic processes, and therefore an understanding of energy is key to understanding how living cells work. Bioenergetics is the study of how organisms manage their energy resources. Remember, energy is the capacity to do work, and it exists in various forms, and cells transform energy from one type into another. So now we're going to talk about various types of energy. First, you have kinetic energy. This is energy associated with the relative motion of objects. Objects in motion can perform work by imparting motion to other matter. Photons of light can be captured and their energy harnessed to power photosynthesis in green plants. And heat or thermal energy is also considered kinetic energy associated with the random movement of atoms or molecules, because when atoms or molecules move, heat or thermal energy is produced. Potential energy is the energy that matter possesses because of its location or structure, meaning it's stored energy. Chemical energy is a form of potential energy stored in molecules because of the arrangement of their atoms. Energy can be converted from one form to another. For example, as a boy climbs the stairs to a diving platform, he is releasing chemical energy stored in his cells from the food he ate for lunch. The kinetic energy of his muscle movement is converted into potential energy as he climbs higher. As he dives, the potential energy is converted back to kinetic energy. Kinetic energy is transferred to the water as he enters it, and some energy is converted to heat due to friction. So here's looking at that in a picture. So as you climb the stairs in the bottom left, we're converting kinetic energy of muscle movement to potential energy. A diver has more potential energy on the platform than in the water. As the diver dives, that potential energy is converted to kinetic energy. And a diver has less potential energy in the water than on the platform. The energy transformations of life are subject to two laws of thermodynamics. So first, thermodynamics is the study of energy transformations. In this field, the term system refers to the matter under study, and the surroundings include everything outside the system. A closed system, approximated by liquid in a thermos, is isolated from its surroundings. In an open system, energy and matter can be transferred between the system and its surroundings. Organisms, obviously, are open systems. They absorb energy, whether it be light or chemical energy, in the form of organic molecules, and release heat and metabolic waste, such as urea and carbon dioxide, to their surroundings. According to the first law of thermodynamics, energy can be transferred and transformed, but it cannot be created or destroyed. So another way of saying that is we usually say is energy can't be created or destroyed. It can only be transformed from one form to another. The first law is also known as the principle of conservation of energy. Plants do not produce energy. They transform light energy into chemical energy. During every transfer or transformation of energy, some energy is converted to heat, which is the energy associated with the random movement of atoms or molecules that I mentioned earlier. A system can use heat to do work only when there is a temperature difference that results in heat flowing from a warmer area to a cooler one. If the temperature is uniform, as in a living cell, heat can only be used to warm the organism. Energy transfers and transformations make the universe more disordered due to loss of usable energy. Here is the first law of thermodynamics at work, where the chemical reactions in the brown bear will convert the chemical, meaning potential energy, in the fish into the kinetic energy, as you will see in the second law of thermodynamics, of running. The second law of thermodynamics states that every energy transfer or transformation increases the entropy of the universe. Entropy is randomness, or a measure of disorder. The more random a collection of matter, the greater its entropy. While order can increase locally, there is an unstoppable trend toward randomness in the universe. Much of the increased entropy of the universe takes the form of increasing heat, which is the energy of random molecular motion. Every energy transfer or transformation increases the disorder or entropy of the universe. So as the bear runs, disorder is increased around its body by the release of heat and small molecules as byproducts of metabolism in the form of carbon dioxide and water. A brown bear can run at speeds up to 35 miles per hour, which is as fast as a racehorse. Living cells unavoidably convert organized forms of energy to heat, a more disordered form of energy. The word spontaneous describes a process that can occur without an input of energy. Spontaneous processes don't have to occur quickly, and some spontaneous processes are instantaneous, such as an explosion. Some are very slow, such as the rusting of an old car. For a process to occur spontaneously, it must increase the entropy of the universe. Processes that decrease energy are non-spontaneous, and they will only occur if energy is provided. Living systems create ordered structures from less ordered starting materials. For example, amino acids are ordered into polypeptide chains. The structure of a multicellular body is organized and complex. For example, animals consume complex molecules in their food and release smaller, lower energy molecules and heat into the surroundings. Over evolutionary time, complex organisms have evolved from simpler ones. This increase in organization does not violate the second law of thermodynamics. The entropy of a particular system, such as an organism, may decrease as long as the total entropy of the universe, meaning the system plus the surroundings, increases. Organisms are islands of low entropy in an increasingly random universe. The evolution of biological order is perfectly consistent with the laws of thermodynamics. The free energy change of a reaction tells us whether the reaction occurs spontaneously. So, in other words, how can we determine which reactions occur spontaneously and which ones will require an input of energy? The concept of free energy provides a useful function for measuring spontaneity of a system. Free energy is the portion of a system's energy that is able to perform work when temperature and pressure is uniform throughout the system, as in a living cell. So when the PowerPoint converted over, for some reason, all of these symbols for delta, which is a triangle, did not appear as a triangle, they appear as a rectangle. So I just tried to fix that. So the free change, the change in free energy, which is called delta G, during a process is related to the change in enthalpy, meaning a change in the total energy, which is called delta H, minus a change in entropy, and entropy is delta S. So delta S means change in entropy. Delta H means change in total energy. And the temperature in Kelvin units. Note that not all energy in a system is available for work because the entropy component must be subtracted from the enthalpy component. What remains is the free energy that is available for work. And this free energy can be thought of as a measure of stability of a system. Delta G is negative when it is spontaneous, meaning when it happens on its own without any additional input of energy. Processes with zero or positive delta G are never spontaneous. And these spontaneous processes can be harnessed to perform work. Free energy is a measure of a system's instability, its tendency to change to a more stable state. During a spontaneous change, free energy decreases and the stability of a system increases. Equilibrium is a state of maximum stability. And a process is spontaneous and can perform work only when it's moving toward equilibrium. Another way to help me remember it is the greater the decrease in free energy, the more work a spontaneous process can perform. So nature runs downhill. As you can see here, when you have a lot of free energy, the system is less stable and there's a greater capacity to do work. As work is done, there's a change, and the free energy of the system decreases. So delta G becomes less than zero. The system becomes more stable, and the released free energy can be harnessed to do work. When there is less free energy, meaning a lower G, the system is more stable and there is less capacity to do work. The concept of free energy can be applied to the chemistry of life's processes. You already know that reactions, chemical reactions, can be classified as either exergonic or endergonic based on free energy. An exergonic reaction proceeds with a net release of free energy, meaning delta G is negative. The magnitude, meaning the quantity of delta G for an exergonic reaction, is the maximum amount of work the reaction can perform. The greater the decrease in free energy, the greater the amount of work that can be done. So for example, the overall reaction of cellular respiration, when we take glucose and we add oxygen and we produce carbon dioxide and water, has a delta G of negative 686 kilocalories per mole. Each mole of glucose, 1 mole of glucose is C6H12O6. Broken down by respiration, 686 kilocalories of energy are released and made available to do work in the cell. An endergonic reaction is one that absorbs free energy from its surrounding. Endergonic reactions store energy in molecules, and delta G is positive. They are non-spontaneous, and the magnitude of delta G is the quantity of energy required to drive that non-spontaneous reaction. So if cellular respiration releases 686 kilocalories, then photosynthesis, the reverse reaction, must require an equivalent investment of 686 kilocalories to convert carbon dioxide and water into sugar. Photosynthesis is strongly endergonic, powered by the absorption of light energy. Reactions in a closed system eventually reach equilibrium and can do no work. A cell that's reached metabolic equilibrium has a delta G equal to 0, and it is dead. Cells are not in equilibrium because they are open systems. The constant flow of materials into and out of the cell keeps metabolic pathways from ever reaching an equilibrium state. A cell continues to do work throughout its life. A catabolic process in a cell releases free energy in a series of reactions, not a single step. Some reversible reactions of respiration are constantly pulled in one direction, as the product of one reaction does not accumulate but becomes the reactant in the next step. Sunlight provides a daily source of free energy for photosynthetic organisms. Non-photosynthetic organisms depend on a transfer of free energy from photosynthetic organisms in the form of organic molecules. ATP powers cellular work by coupling exergonic reactions to endergonic reactions. And a cell does 3 main types of work. First, you have mechanical work, such as the beating of cilia, the contraction of muscle cells, and the movement of chromosomes during cellular reproduction. You also have transport work, the pumping out of substances across a membrane against the direction of spontaneous movement, and chemical work, like driving endergonic reactions, such as the synthesis of polymers from monomers. Cells manage their energy resources to do this work by energy coupling. Energy coupling is the use of an exergonic process to drive an endergonic one. In most cases, the immediate source of energy to power cellular work is ATP. Adenosine triphosphate is a type of nucleotide consisting of the nitrogenous base adenine, the sugar ribose, and a chain of 3 phosphate groups. The bonds between the phosphate groups can be broken by hydrolysis. Here's a picture of the ATP. You can see the adenine, nitrogenous base, the ribose sugar, and the 3 triphosphate groups. The bonds between the phosphate groups of ATPs can be broken by hydrolysis. Energy is released from ATP when the terminal phosphate bond is broken. This releases 7.3 kilocalories of energy per mole of ATP under standard conditions. This release of energy comes from the chemical change to a state of lower free energy, not from the phosphate bonds themselves. So you take adenosine triphosphate, undergoes hydrolysis, the addition of water, and what that does is that breaks off an inorganic phosphate, so it has a diphosphate now, and releases the energy. Why does the hydrolysis of ATP yield so much energy? Each of the three phosphate groups has a negative charge. These three like charges are crowded together, and their mutual repulsion, because like charges repel, contributes to the instability of this region of the ATP molecule. In the cell, the energy from the hydrolysis of ATP is directly coupled to endergonic processes by the transfer of a phosphate group to another molecule. So 3 types of cellular work, the mechanical, the transport, and the chemical that we spoke about earlier, are powered by the hydrolysis of ATP. And as I just said, in the cell, the energy from the exergonic reaction of ATP hydrolysis can be used to drive an endergonic reaction. Overall, the coupled reactions are exergonic. So in each case, a phosphate group is transferred from ATP to another molecule, and the phosphorylated molecule undergoes a change that performs work. The recipient molecule is now called a phosphorylated intermediate. In A, you see that glutamic acid is converted to glutamine. This by itself is an endergonic reaction. So delta G is positive. It is not spontaneous. In B, we see glutamine synthesis occurs in 2 steps. First, ATP phosphorylates glutamic acid, making it less stable with more free energy. And secondly, the ammonia displaces the phosphate group, forming glutamine. In C, free energy change for coupled reaction. Here you have the delta G for the glutamic acid conversion to glutamine, which is 3.4 kilogals per mole, plus the delta G for the ATP hydrolysis, negative 7.3 kilocalories per mole. Gives the free energy change for the overall reaction as negative 3.9 kilocalories per mole, because the overall process is exergonic. There is a net delta G, which is negative, so this occurs spontaneously. Transport and mechanical work in the cell are also nearly always powered by the hydrolysis of ATP. In these cases, ATP hydrolysis leads to a change in a protein shape and often its ability to bind to other molecules. Sometimes this occurs via a phosphorylated intermediate, as seen for the transport protein that we just spoke about. In A, you can see the ATP hydrolysis causing a change in the shape and binding affinities of proteins directly by phosphorylation. So here you see the membrane protein carrying out active transport of a solute. In B, we see it being done indirectly via non-covalent binding of ATP and its hydrolytic products, as in the case for motor proteins that move vesicles and other organelles along the cytoskeletal tract. ATP is a renewable resource that can be regenerated by the addition of a phosphate group to ADP. The energy to phosphorylate ADP comes from catabolic reactions in the cell. A working muscle cell recycles its entire pool of ATP once each minute, and more than 10 million ATP molecules are consumed and regenerated per second per cell. That's a lot. Regeneration of ATP is an endergonic process requiring an investment of energy. Here you can see energy released by breaking down, or the catabolism of a reaction in a cell is used to phosphorylate ADP, which regenerates ATP. The chemical potential energy stored in ATP drives most cellular work. A catalyst is a chemical agent that speeds up the rate of a reaction without being consumed by the reaction, meaning it can be used over and over again. An enzyme is a catalytic protein, and enzymes regulate metabolic pathways. Every chemical reaction involves bond breaking and bond forming. For example, to hydrolyze sucrose, the bond between glucose and fructose must be broken and new bonds must form with hydrogen and hydroxyl ions from water. So here you can see sucrose being hydrolyzed. To reach a state where bonds can break and reform, a reactant molecule must absorb energy from their surroundings because you need energy to break bonds. When the new bonds of the product molecules form, energy is released as heat, as the molecules assume stable shapes with lower energy. The initial investment of energy for starting a reaction is the free energy of activation, or sometimes better known as activation energy, capital E with a smaller capital A. Activation energy is the amount of energy necessary to push the reactants over an energy barrier so that the reaction can proceed and products can be produced. Again, the activation energy may be supplied in the form of heat that the reactant molecules absorb from their surroundings. The bonds of the reactant molecules break only when the molecules have absorbed enough energy to become unstable and therefore more reactive. This is called a potential energy diagram, and this is of an exergonic reaction. This is a reaction in which you have a negative delta G. I know this by looking at the diagram because my products are lower than my reactants. You can see the reactants on the left-hand side. You have A bonded to B, C bonded to D. In order to get to that transition state, or intermediate at the top of the hill, we need activation energy. It's at that point where the bonds between A and B and C and D can break and new bonds can form, resulting in the products of AC and BD. Here you can see the difference with how the reaction would proceed without an enzyme. You can see in black that the intermediate is much higher, and therefore the activation energy required without an enzyme is almost double than it would be with an enzyme. You can also see that where the reaction starts at the reactant line and the product line, they remain the same with or without the catalyst. So the only thing that is different is going to be delta H, the overall energy for the reaction. One very important thing about enzymes is that they are substrate specific. The reactant that an enzyme acts on is the substrate. The enzyme binds to its substrate, forming an enzyme-substrate complex. While the enzyme and substrate are bound, the catalytic action of the enzyme converts the substrate to the product or products. The specificity of an enzyme results from its three-dimensional shape. While only a portion of the enzyme binds to the substrate, the portion that does, or the active site of the enzyme, is typically a pocket or groove on the surface of the protein into which the substrate fits. The active site is usually formed by only a few amino acids, and the specificity of the enzyme is due to the fit between the active site and the substrate. As the substrate enters the active site, interactions between the substrate and the amino acids of the protein causes the enzyme to change its shape slightly, leading to a tighter induced fit that brings chemical groups in position to catalyze the reaction. Here you can see a picture of the substrate. You can see a picture of the active site, and when it fits right in there, the enzyme substrate complex is formed. In most cases, substrates are held in the active site by weak interactions, such as hydrogen bonds and ionic bonds. Enzymes are extremely fast acting and emerge from the reactions in their original form. Very small amounts of the enzyme can have a huge metabolic effect because they are repeatedly used over and over again in catalytic cycles. Here you can see the two substrates entering the enzyme, and they are held in there by weak interactions. That you can see when this happens that the active site lowers the activation energy and speeds up the reaction. The substrates are then converted to products and they are released. The active site is now available again on the enzyme to receive more substrates. Enzymes use a variety of mechanisms to lower the activation energy and speed up the reaction. In one way, if there's more than one reactant, the active site brings substrates together in the correct orientation for the reaction to proceed. Another way, as the active site binds to the substrate, it may put stress on the bonds that must be broken, making it easier for the reactants to reach the transition state. A third way is the R groups at the active site may create a microenvironment that is conducive to a specific reaction. Or an active site may be a pocket of low pH, facilitating hydrogen ion transfer to the substrate as a key step in catalyzing the reaction. Enzymes may briefly bind covalently to substrates. Subsequent steps of the reaction restore the R groups within the active site to their original state. The rate that a specific number of enzymes convert substrates to product in part depends on substrate concentrations. At low substrate concentrations, an increase in the substrate concentration speeds up binding to available active sites. However, there is a limit to how fast a reaction can occur. At high substrate concentrations, the active sites on all enzymes are engaged, meaning that the enzyme is saturated. The rate of the reaction is determined by the speed at which the active site can convert substrate to product. The only way to increase productivity at this point is to add more enzyme molecules. A cell's physical and chemical environment affects enzyme activity greatly. The activity of an enzyme is affected by general environmental conditions such as temperature and pH. It can also be affected by chemicals that specifically influence the enzyme. Temperature has a major impact on reaction rate. As temperature increases, collisions between substrates and active sites occur more frequently because molecules move more rapidly. As temperature increases further, thermal agitation begins to disrupt the weak bonds that stabilize the protein's active conformation, and the protein denatures. It breaks down. Each enzyme has optimal temperature. Most human enzymes have optimal temperatures of about 35 to 40 degrees Celsius. Bacteria that live in hot springs contain enzymes with optimal temperatures of around 70 degrees Celsius or above. So as you can see, depending on the environment, will dictate the optimal temperature. Each enzyme also has an optimal pH. Maintenance of the active conformation of the enzyme requires a particular pH. This falls between pH of 6 and 8 for most enzymes. However, digestive enzymes in the stomach are designed to work best at a pH of 2, while those in the intestine have an optimum pH of 8. Many enzymes require non-protein helpers called cofactors for catalytic activity. Cofactors bind permanently or reversibly to the enzyme. Some inorganic cofactors include zinc, iron, and copper. Organic cofactors are called coenzymes, and many vitamins are coenzymes. Binding by inhibitors prevents enzymes from catalyzing reactions. If inhibitors attach to an enzyme by covalent bonds, inhibition may be irreversible. If inhibitors bind by weak bonds, inhibition may be reversible then. Some reversible inhibitors resemble the substrate and compete for binding to the active site. These type of inhibitors are called competitive inhibitors. Competitive inhibition can be overcome by increasing the concentration of the substrate. Non-competitive inhibitors impede enzymatic reactions by binding to another part of the molecule. Binding by the inhibitor causes the enzyme to change its shape, rendering the active site either less effective or non-effective at catalyzing the reaction. Toxins and poisons are often irreversible enzyme inhibitors. Sarin is a nerve gas that was released by terrorists in the Tokyo subway in 1995. Sarin binds covalently to the R group on the amino acid serine. Serine is found in the active site of acetylcholinesterase, an important nervous system enzyme. At the top, you can see a substrate combined normally to the active site of an enzyme. In B, when you have a competitive inhibitor, it mimics the substrate, competing for the active site. So now the substrate cannot bind. A non-competitive inhibitor, as in the last diagram, binds to the enzyme in a way that it is not at the active site. It's somewhere else on the enzyme, but it alters the shape of the enzyme so that even though the substrate can still bind, it cannot bind effectively. Or, depending on if the shape is changed too much, it won't be able to bind at all. Enzymes are proteins encoded by genes. Changes or mutations in genes lead to changes in amino acid composition of an enzyme. Altered amino acids, particularly at the active site, can result in novel enzyme activity or altered substrate specificity. Under environmental conditions where the new function is beneficial, natural selection would favor the mutated allele. For example, repeated mutation and selection on the B-galactosidase enzyme in E. coli resulted in a change of sugar substrate under lab conditions. In other words, researchers tested whether the function of the enzyme B-galactosidase, which breaks down sugar lactose, could change over time in populations of the bacterium E. coli. After 7 rounds of mutation and selection in the lab, the B-galactosidase evolved into an enzyme specialized for breaking down sugar other than lactose. This ribbon model shows one subunit of the altered enzyme. 6 amino acids were different. 2 changed amino acids were found near the active site, and 2 changed amino acids were found on the surface, and 2 were found in the active site. This simplified model is generally accepted as the main way in which a multitude of different enzymes arose over the past few billion years of life's history. Chemical chaos would result if a cell's metabolic pathways were not tightly regulated. A cell does this by switching on or off the genes that encode specific enzymes by regulating the activity of enzymes. In many cases, the molecules that naturally regulate enzyme activity behave like reversible non-competitive inhibitors. Regulatory molecules often bind weakly to allosteric site, a specific receptor on the enzyme, away from the active site. Binding by these molecules can either inhibit or stimulate enzyme activity. Most allosterically regulated enzymes are constructed of 2 or more polypeptide chains. Each subunit has its own active site. Allosteric sites are often located where subunits join. The binding of an activator stabilizes the conformation that has functional active sites, while the binding of an inhibitor stabilizes the inactive form of the enzyme. As the chemical conditions in a shell shift, the pattern of allosteric regulation may shift as well. So in the upper left, you see an allosteric enzyme with 4 subunits. You can see the active site is like a half moon. And then you see the regulatory site of one of the 4, which is the V section. An activator, an allosteric activator, which is the green molecule, stabilizes the active form so that it doesn't move by fitting in to that regulatory site. The enzyme can switch or oscillate between the active form and the inactive form, and you can see what the non-functional active site looks like. An inhibitor can fit into that non-functional active site and stabilize the inactive form. In enzymes with multiple catalytic subunits, binding by a substrate to one active site stabilizes favorable conformational changes at all other subunits. This is known as a process called cooperativity. This mechanism amplifies the response of enzymes to substrates, priming the enzyme to accept additional substrates. Cooperativity is allosteric because binding by a substrate to one active site affects catalysis in a different active site. Here you can see the inactive form on the left oscillates with the active form on the right when the active form is not stabilized by the substrate. The binding of the substrate molecule to the active site on one subunit locks all the other subunits into active conformation. A common method of metabolic control is feedback inhibition, in which an early step in the metabolic pathway is switched off by the pathway's final product. The product acts as an inhibitor of an enzyme in the pathway. Feedback inhibition prevents a cell from wasting chemical resources by synthesizing more product than is needed. As you can see here in this example of feedback inhibition, some cells use this 5-step pathway to synthesize the amino acid isoleucine from threonine, another amino acid. As isoleucine accumulates, it slows down its own synthesis by allosterically inhibiting the enzyme for the first step of the pathway. Feedback inhibition thereby prevents the cell from making more isoleucine than is necessary and thus wasting chemical resources. Structures within the cell help bring order to metabolic pathways. A team of enzymes for several steps of a metabolic pathway may be assembled as a multi-enzyme complex. The product from the first reaction can then pass quickly to the next enzyme until the final product is released. Some enzymes and enzyme complexes have fixed locations within the cells as structural components of particular membranes. Others are confined within membrane-enclosed eukaryotic organelles. For example, enzymes for cellular respiration are located in the mitochondria. Here you can see the matrix contains enzymes in solution that are involved in the second stage of cellular respiration. Enzymes for the third stage of cellular respiration are embedded in the inner membrane. So as you can see, some enzymes are grouped into complexes, some are incorporated into membranes, and some are contained inside organelles, increasing the efficiency of all metabolic processes. And this ends our lesson on Chapter 8.